Hi everybody, this is Mrs. Baker and I'm going to lead you through your notes on bacteria. Remember to leave enough space to write your questions and your summary. Our title is Bacteria, and our essential question is, what organelle does a bacterium lack? In other words, doesn't have. What organelle does a bacterium lack? There you go. That differs from a eukaryote. So how is a bacteria different from a eukaryote? What does it not have? Bacteria are prokaryotes, which are unicellular, one-celled, unicellular organisms that lack a nucleus. So that answers our first question. They lack a nucleus. Typically, they range in size from 1 to 5 micrometers. Micrometer is 1/millionth of a meter, so these are really, really tiny cells. They are identified by characteristics such as shape, chemical nature of their cell walls, and the way they move, and obtain energy. So how do they get their food, their nutrients? The different shapes of prokaryotes are bacilli. A bacillus, that's the singular, is a little rod. So they look like little sausages, sort of. Sometimes they even make little links together. So these are our rod-shaped bacteria. There are others called cocci, C-O-C-C-I, and these are spherical. In other words, they look like little spheres or circle. Sometimes they get into little clumps. Sometimes they make little chains of circles. Sometimes they're by themselves. And then there are some called spirilla, which look like little corkscrews or little spirals. These are spirilla. Spiral or corkscrew shaped. We also look at the cell walls to classify bacteria into different groups, and there is a test called a Gram test or Gram stain that we can use. Gram staining is a testing method that determines if bacteria's cell walls have peptidoglycan, which is a compound that some bacteria have, but others don't. And basically they put chemicals on the bacteria, and depending on what color they turn, they can tell if they're gram-positive or gram-negative. In other words, they have peptidoglycan or not. We also look at the movement of bacteria. Some bacteria are propelled or moved forward by a structure called a flagella, which is a whip-like structure, or whip-like structure. This is a bacterium. It kind of looks like a little tail, a little whip at the end. You can't see my little flagella there. There's a little flagella. Others move by secreting a slime. By secreting slime. So they kind of slide around on this little slime that they put out. And others, or they may be carried away by the air or water. So they go around through nature just being carried away in the flow of things. We look at their food and energy intakes also to classify them. Some bacteria are autotrophs. So some bacteria make food using the sun. That's called photosynthesis. Not just plants do it. Some bacteria do it too. Using sun, or others are chemotrophs. They can use chemicals to make their own food. But other bacteria are heterotrophs. Some are autotrophs, some are heterotrophs, because they need to take in food, take in nutrients by consuming other organisms. So they will eat other organisms or food made by other organisms. When you ingest or you have a bad bacteria in your body, it is destroying your cells because it's using them for nutrients. I'm going to grab a card because I am out of space here. So go ahead and get one of those from your teacher so that we can write the last part, which is about reproduction. Bacteria can reproduce through a process called binary fission, which is like mitosis, where you make two identical cells. So it's a reproductive process that involves only one parent cell. So this is asexual reproduction, and results in two identical cells. Then there's conjugation, which is where a hollow bridge is formed between two bacteria, and genes flow from one to the other. Basically, they just give each other a piece of the DNA through this little tube that forms between them. And then some bacteria can form endospores. And I'm going to run out of room here. Hopefully you write a little bit smaller than me, so I'll see how much I can fit in here. An endospore is a spore that is formed. It's kind of like a seed, but it's not really a seed. I don't like to call it that, but kind of think about it like that. It's a spore that is formed when a bacterium produces a thick internal wall that encloses its DNA. this up here and bring another card for myself so that we have enough room. That encloses its DNA, and the cell will remain dormant, or kind of like in hibernation, until more favorable growth conditions are present. So basically the cell shuts itself down, gets in a hard little shell, and is able then to stay essentially dormant for a while.